Hi, welcome to our third lecture on the cell division chapter. This one deals primarily with meiosis. So to begin with, let's talk about what meiosis produces. Being specific in terms of the number, type, and genetics of the cells being produced, you'll end up with 4 haploid, genetically unique gamete cells. This is different than mitosis, where you have 2 diploid, genetically identical somatic cells. So today we're going to go over meiosis in detail. It's a very pivotal lesson because a couple of things that happen during meiosis are crucial for evolution to occur. So that's why meiosis is a very, very important type of cell division. Okay, let's get started. So if nothing else, know this about meiosis. It occurs in germ cells, which are also known as reproductive cells, and that's it. Every other cell would be considered a somatic cell and is going to undergo mitosis. The parent cell, the parent germ cell, is going to go through 2 rounds of division. And therefore it's going to result in 4 daughter cells instead of 2. These daughter cells are called gametes, and that is a classification for either egg or sperm, depending on the gender. Each gamete is genetically unique with only half the amount of chromosomes. That's known as a haploid. Gametes will combine. On their own, having a haploid number of chromosomes will do very little for the successful ability to live. But when you combine them, when 2 gametes combine, such as an egg and a sperm, that will give you a diploid set, a full set of chromosomes. That initial combination is known as a zygote. And a zygote undergoes mitosis to become an embryo becomes a fetus, and a fetus becomes an organism. Okay, so if you know this, that's great. You know what meiosis does, but not so much how it does it. So the rest of this presentation is really about how meiosis goes about accomplishing these things. Okay, before we begin, let's look at a few key vocabulary words. First, haploid and diploid. Well, you may know these. Haploid means a cell with half the amount of chromosomes. Diploid means a cell with a full set of chromosomes. You may know that already, so that's not that big of a deal. What's really important here is the difference between a gene and something called an allele. A gene, we looked at genes earlier during the DNA chapter, and we defined them as a sequence of nucleotides that codes for a trait. And that's pretty much the same definition we have here. Here we have a segment of DNA that codes for a trait. An allele is a little bit more specific. It's slightly different than a gene. An allele is an alternate version of the same gene. So, for example, this is the best way to describe the difference between these two. For example, a gene would be hair color, and then an allele would be the actual hair color. A brunette, a blonde, a redhead, dark hair, black hair, that kind of thing. A gene for another gene might be eye color, and then the alleles would be blue, brown, hazel, green, that sort of thing, that sort of description. So when you hear the phrase, you have really good genes, or you're going to inherit really good genes, that's not really accurate because everyone has the same genes. Everyone has the hair color gene. Everyone has the eye color gene. Everyone has the hair gene. It may be turned off, but you still have that gene. What's different about people are the alleles that they inherit. So while everyone inherits the same genes, people will inherit different alleles. So let's talk about that. Let's take a picture of what that may look like. You have 23 pairs of chromosomes. One is from mom and one is from dad. So the chromosomes can actually be identified based on their length. And usually the longer the chromosome, the lower the number it'll appear on the number of chromosomes that you have. So here we have our 22 chromosomes listed out, and then our sex chromosomes, which determine our gender. The longest one is going to be over here, and then the shortest ones are down here. Each pair has the same gene. So each one of these pairings has the same genes, but they may have different alleles because one of them came from your mom and one of them came from your dad. So again, this is from your mom, this one over here, that one's from your dad. Mom, dad, all the way down. Same genes, but possibly and most likely different alleles. If you were to expand on that, let's take a look at chromosome number 5. Here, we've broken apart, and then this sequence of DNA that's compacted here, that would represent one full gene on this chromosome that you got from your mom. Over here, you have the same gene, but notice how the letter is a lowercase as opposed to an uppercase. That's going to represent having different alleles. Same gene, we'll say the gene for ear hair, same gene, but different alleles. The ear hair gene, here your mom has no ear hair, and here your dad has plenty of it. Same thing down here. Here's another gene. Well, let's call this the gene for, I don't know, how much stomach acid your stomach produces. Here, one allele says you produce a lot, and one allele says you produce very little. Our next gene down turns out to be the same allele. So let's say this is the gene for eye color, and a big C represents blue eyes, and this big C, that also represents blue eyes. So your mom, the allele from your mom will be the same allele from your dad, so you're going to be guaranteed to have blue eyes. That's not exactly how it works, but it's okay for the simplification of what we're learning right now. We'll get into more detail with that in the inheritance chapter. One important thing to note is that these are not sister chromatids. Sister chromatids are exactly the same in every way, shape, and form. These are something known as homologous chromosomes. So again, sister chromatids are chromatids that have identical genes and alleles, but they're counted as one chromosome because they're the same thing. Homologous chromosomes are chromosomes that have identical genes, but the alleles may be different. And that's because one of the chromosomes came from your mom and one of them came from your dad. And because these alleles are not identical, they're counted as a pair of chromosomes or as two chromosomes. Understanding the difference between sister chromatids and homologous pairs is paramount to understanding the difference of, or understanding how meiosis works. It's super, super critical. So make sure you fully understand what a sister chromatid looks like and what a homologous chromosome looks like. Another giveaway is that sister chromatids are always attached by a centromere, so they're kind of bow tied together. Homologous pairs are not. They're just near each other and they look similar. Here's a visual. It's not as good as ones that you can get online, but it's something that I made that looks okay. Here's a sister chromatid. This chromatid will have replicated itself in every way, shape, or form. Same colors, same genes, same alleles. Here's a homologous pair. Notice that the colors are the same. So green, green, blue, and blue, and yellow, and that yellow. That's supposed to represent similar genes, the same gene, but different alleles. When you have a pair of homologous sister chromatids, you have what is called a tetrad. That's going to be important for the first couple phases of meiosis. Keeping track of chromosomes and chromatids. Again, here this is one chromatid, also one chromosome. Here we have, when it duplicates itself, because it's attached, it's still considered one chromosome, but it's two chromatids. Another way to say this is a double-stranded chromosome, or a sister chromatid. When this splits, when the chromatids are no longer attached, you can now start counting them as their own individual chromosome. Makes it a little, it's complicated, but that's kind of how the counting of chromosomes occurs. Okay, one thing that all homologous chromosomes are going to do before cell division, whether or not it's mitosis or meiosis, is they're going to copy themselves and make sister chromatids. So if you look at this pairing here, pair number 1, each one of these chromatids is going to copy itself and give itself a sister chromatid, and that shows up right here. So here's pair number 1, but if you notice here, there's 4 total chromatids. 1, 2, 3, 4. The ones that are linked together are identical copies, and then if you compare them to each other, they would be considered homologous pairs. So whether or not it's mitosis or meiosis, this is always going to happen. In mitosis, these sister chromatids are going to get in one straight line along the cell's equator, and then one chromatid will go one way, the other identical chromatid will go the other way, and you'll result with 2 nucleuses with the exact same genetic information. That's not exactly how they line up during meiosis, and that results in having genetically unique cells. I'll show you what I mean in just a second. By the way, this is just a picture of a human karyotype. A karyotype is a picture of all of your genes laid out. So here's a fly karyotype. Notice that they only have 8 chromosomes, whereas a human has 46. Okay, the cell cycle of a germ cell. A germ cell is going to go through most of the same thing that a somatic cell is going to go through. It's going to go through G1, S phase, and G2, and when it's ready to divide, it's going to go through M phase. However, this M phase is different in that there's a meiosis 1 and a meiosis 2. So these 2 rounds of division is what allows for the cell to get down to half the amount of genetic information. Once again, in interphase, the cell DNA will copy itself, so you'll see 2 red chromatids and 2 blue ones, 2 green, and 2 yellow. Okay, so this is just kind of prepping us for, I'm going to walk you through the 8 stages of meiosis, and instead of using 46 chromosomes as you would in a human. I'm only going to use 8 just so we can kind of simplify things and keep track a little bit more easily. Remember, right here up in our original number of chromosomes, this organism will have 8 chromosomes or 4 pairs of homologous chromosomes. During S phase, they'll duplicate, so you'll have sister chromatids here and here, and then together this whole thing will be called a tetrad. And another way to describe a tetrad is a homologous pair of sister chromatids. Okay, quick review. Here's homologous pair, same chromosomes but different allele, same genes but different alleles. These two are exactly identical, so they'll be known as sister chromatids, and then together a homologous pair of sister chromatids is known as a tetrad. Prophase 1. During prophase 1, the sister chromatids are attached via a centromere. The homologous chromosomes begin to find each other. So this is a homologous pair with this one, and the blues are going to try to find each other, the yellows, the oranges, and the red, and the greens, they're going to try to find each other. When they find each other, the resulting structure is called a tetrad. When they're close to each other, something really important is going to happen. This process called crossing over is going to occur. Basically, what's going to happen is the leg of one chromosome is going to cross over with its homologous pair and literally exchange segments of the chromosome, so that a little bit of this one ends up over here, and a little bit of, we'll say, this one ends up over there. Basically, this is going to ensure that your offspring will be a blend of your parents. One quick note about crossing over, which is hugely important, is that alleles, alleles will be, the alleles from homologous chromosomes are going to trade places. And what's going to result, so if you look down here, this is a sister chromatid, and this is a sister chromatid, but when they combine, you can't really call them sister chromatids anymore because they've recombined, so they're no longer identical. We call that recombinant chromosomes or recombinant DNA because it's recombined. Not the biggest detail, but it's something that is worth pointing out anyway. All right, in metaphase 1, the tetrads will align in the middle, and the way that they line will be random, such that some of these darker chromosomes, let's say the darker ones are your mom's chromosomes and the lighter ones are your dad's. It's not like all of your mom will line up on one side and all of the dad will line up on the other side. It's going to be a mixture, a random assortment of that. And that process is actually called independent assortment. And that also, just like crossing over, kind of shuffles where the genes end up, thus ensuring that your offspring will be a mixture of your parents. Anaphase 1, the homologous pairs will separate, so the tetrad is no longer there. It's kind of disappeared. The sister chromatids are still tied together with a centromere, though. That's important. But again, they're not really sister chromatids because of crossing over. In telophase 1, the cytokinesis will create 2 new daughter cells, each with that recombined sister chromatids. Okay, a little checkpoint. How many cells are there after meiosis 1? There's 2. Are the number of chromosomes haploid or diploid? Well, it kind of depends. At this point, if you count the sister chromatids as one chromosome, which you normally would do if they're attached through a centromere, then it's haploid. If you count them as 2 chromosomes, because you kind of can because they're not exactly sister chromatids anymore, then I guess you can consider them diploid. What major events happen during meiosis 1? That would be crossing over and independent assortment, and they ensure that each gamete produced will be a genetically unique combination of your mother's and your father's genes. Okay, I need to take a time out for the bigger picture of the life science unit. The whole purpose of the life science unit is to give you everything you need to know about how evolution works from down to the very molecular level, and that's kind of where we are right now with studying cell division. Evolution is based off of genetic variation. If we were all clones and we got a virus, all of us would respond to the virus in the exact same way. And if it was a dangerous enough virus, then the entire species would get wiped out. However, if your species is genetically diverse, that means that you may be able to respond to an environmental pressure, such as a virus, differently than other members of your species. And maybe you might respond to it a little bit better, and you'll be allowed to live, or you'll overcome the virus. So having that variation is crucial for a species' ability to adapt. And the more diversity you have, the more diversity a species has, the more likely it is to adapt. There are 4 ways that biology encourages genetic diversity. 2 of them occur during meiosis. Crossing over, oops, I'm going to need this right here, crossing over and independent assortment. Number 3 and number 4 we'll get to in a couple of weeks, but for now let's just be concerned with how crossing over and independent assortment encourage the species to have genetic diversity. Remember this slide, you're going to see it again when we go over 3 and 4. Okay, back to meiosis. So the great thing about meiosis too is that it's basically the exact same thing as mitosis. So it's a good thing you already know how that works. The only difference is that instead of starting with pure sister chromatids, you're going to be starting with recombined chromosomes. So they're pretty similar, but there might be some segments that are slightly different than its pair. So metaphase 1, the quote-unquote sister chromatids will line up in the middle. Anaphase 1, they'll get pulled apart to the ends by the centromeres and the mitotic spindle. Telophase 1, the nuclear envelope will reappear, and then cytokinesis will cut it into 2 different cells. If you compare the gametes that are created with the original cell, you'll notice that there's half the amount of genetic information. So there's 8 chromatids here, and then 4 in each cell. So they have half the number of chromosomes, which makes them haploid. If you compare them to each other, you'll notice that none of them are identical. So you have genetically unique gametes. This also ensures that you won't have children that are identical to each other. So just as a review to close this out, meiosis 1 and meiosis 2 are the 2 phases of nuclear division for germ cells. During meiosis 1, crossing over and independent assortment mix up parental DNA, and the homologous chromosomes are pulled apart. During meiosis 2, sister chromatids are pulled apart, and it results in 4 genetically unique haploid gametes. Okay, that's it for meiosis. If you have any questions, please come in and let me know, and we'll work through them. Thanks so much.